and we talked about the microbial techniques that we have, how to culture microorganisms. We talked about how to preserve microorganisms. I could remember some like I could remember we talked about cryopreservatives, cryopressants or something. I can't really remember my then um just microbiology generally, the application of microbiology in our world today. So and that's all I could remember.
Yeah,
Yes.
thank you. You've done well. All right. Who else wants to try?
Okay, good morning, ma.
Good morning, dear.
You talked about the history of microbiology,
Okay.
the biologists. I remember us talking about pasteurization. We talked about sterilization too. Yeah, the cryoprotectants and other microbiology in general, the areas of microbiology, different areas where you can use microbiology. Yeah, that's what I can remember.
Okay. Thank you very much. The reason I quickly want you to think back is because when I start mentioning words like techniques, preservation, cultures, dilutions, you will not be thinking, what is the meaning of this? Thank you. So we'll move on quickly. So
Good morning, ma.
good morning there. God bless you. So I've introduced myself, Dr. O.M. Braimon, Department
Yes,
of
ma.
Microbiology. Yes?
Please, ma. It's not everybody that do microbiology in year one. So I remember you're trying to say that it's not everybody that will be able to get to know what you're saying
No
because
problem. But
we've
at
not
this
done it.
level, with 3 weeks lecture, you won't be able to go back to 100 level work, right? So, but as much as possible, there's no big deal. You are going to understand everything we are talking about. So when you are reading and there's a strange word that you don't understand, what do? You find out, right? You know, a teacher cannot pick every word one by one in the class for a 1 or 2 hours lecture. So today we are starting with topic 1. I'm switching off my video because I want to have a good network. I don't want any disruption. So you can do likewise, because if you put on your video, then it might disrupt your internet connectivity. So we are talking about today, topic 1, preservation techniques of laboratory cultures. Apart from this first topic, I will also be teaching you microorganisms in relation to food. What is the work of or the purpose of microorganisms in food? What role do they play in food? The same thing, what role do microorganisms play in the industries and in our environment? Then finally, we'll look at microbial variation and heredity. We'll look at the 3 laws of variation and heredity. The laws of heredity, the Median law, Mendelian law, which you have already learned in your secondary school, and then I'll refresh your memory. Then we look at the Punnett square. So this is all I have to teach you for 2 weeks. And I know we can do it. If we do 2 hours today, then we do 1 hour on Friday. The next week, Tuesday, we can do 2 hours also. And then on Friday, we are done. So at the end of this lesson today, you must know how to explain the importance of microbial culture preservation. You must know why. Why are you trying to preserve microbial culture? You must be able to identify different preservation techniques used in the laboratories. You should be able to describe the advantages and disadvantages of each preservation method that you are learning today. And you should be able to understand the factors influencing the success of microbial preservation. You all know that your exam format for MIC 211 is always CBT, objective. So you need to know to have to play, to take a close look at your slides. So microbial cultures are very necessary for biological or medical or environmental or industrial applications. So whether you are working in the medical lab, in the food industry, in the pharmaceutical industry, or you're a teacher like myself, or a researcher, whether it is oil industry, wherever field, you will make use of microbial cultures, and there will always be microbiology units or departments. The medicals want to find out, okay, so this person is sick, what is responsible, what kind of microorganism is responsible for this ailment? They want to have a look at it. So the industry too want to look at, okay, maybe they are producing beer, or they are producing, please mute yourself before I log you out. You want to look at what organism is going to be okay for their fermentation and all that. So they will have cultures. That is, they will have microorganisms stored somewhere for use. So laboratory cultures, that is aggregate of microorganisms that you have grown. You all know that microorganisms cannot be seen with the naked eye. And you cannot see them until you culture them, you grow them. So after growing this microorganism, whether from medical sample like blood or urine or stool, or from environmental sample like water or soil, or from plant sample, or from any sample, even if it is gala or water, you have to culture these organisms to be able to see them on plates, like you can see below my slide here. And then you now pick some of these colonies to be able to observe under the microscope. So laboratory cultures play a very crucial role in scientific research, whether it is in the research industry or medical or industrial applications. Laboratory cultures also provide a stable and reproducible environment for studying microorganisms, their cells, and other biological entities of those microorganisms. We want to study the cells because microorganisms differ. So the shapes of the cell, the texture, the color will help you or guide you to determine what kind of microorganism is this. And apart from studying the microorganisms themselves, you want to study other biological entities, maybe their product, their metabolic product, or their enzymes. Then you want to maintain microbial viability. If I'm working in a research industry and I carry out research, maybe for pharmaceutical purpose or for food industrial purpose, I cannot be going to isolate organism each time I want to carry out my fermentation or I want to produce my product. So as soon as I have a good microorganism, I want to maintain them. I want to keep them. I want to prevent them from dying. So that is what it meant by maintaining microbial viability. You make it viable. You make them to be alive. You keep them alive, and you make their genetic components stable over a long period of time. So if you don't use the appropriate preservation techniques, you will lose those microorganisms. They will die after a while. I still have the organisms that I isolated since 2008. I still use them up till now, and I'm using them for further works. I carry them up and down. I carry them to the US. I carry them to Germany. I carry them to wherever I go to carry out studies. So you have to determine, you have to employ different storage or preservation techniques to determine which method suits your organisms best. So these methods aim to maintain the genetic and phenotypic, that is the genetic or phenotypic, that is the physical character. You will still learn about phenotypic character later in this part of the study. So you can maintain the genetic components so that they do not alter, and then you can preserve the phenotypic, that is the physical characteristics of the organism over a long period of time. And this allows researchers to retrieve and study them when needed. Like I said, you don't have to go to the environment or collect food or blood samples each time you want to carry out your analysis. When you store your organisms, you can also go back to wherever you have stored them and continue your studies from where you store. Several preservation techniques, are available. So, what are the objectives of microbial preservation in summary? To maintain microbial viability and activity, to prevent contamination. If you don't preserve your organisms very well, they will be contaminated. That is, you will have unwanted microorganisms growing along with the organisms that you want to use. Then to retain genetic stability, because you don't want your organisms to mutate. When they mutate, they drift from their usual genetic characteristics that enable you to do whatever you want to do with them, and then you will be having something else. So to prevent mutation, to have genetic stability, and then to enable long-term storage, storing microorganisms for extended period without losing their essential characteristics. Now, I told you the one I've saved since 2008, at least I have 9 of them. I lost one, so I have 8 still alive. I lost one, I think around maybe 2015 or 16 or thereabout. Because before I realized that the method I use in storing others did not work for that one. It was already dead. So, and we'll now look at the first method. The first method, you can use periodic subculturing. This is one method that you can use. What does this mean? It involves transferring organisms to fresh growth media at regular interval. By now, you should have learned about media and all that. So you can imagine if you grow organism, you prepare media, you grow organism, then after 3 months, you go back again, prepare another media, and transfer this organism. You keep doing that at regular interval after studying your organism. How frequently do you need to change the growth media? Sometimes it is every 3 months, sometimes it is every 6 months, sometimes it is every year. So, and this method can be used for bacteria, fungi, and yeast. It is a simple method. That's the advantage. It is also not expensive, but it is very labor intensive. You can imagine if you have 100 isolates or microorganisms, and every 3 or 6 months, you have to be preparing media, 100 media to resubculture these isolates. And each time you bring the microorganisms out, you are taking a risk of contamination because your environment might be contaminated at that particular time. And then there's the likelihood for genetic changes over time. So that is the disadvantage of periodic subculturing as a method of preservation of culture. Refrigeration is another method. And refrigeration is a short-term storage method. For refrigeration, it is simple and it is what we widely use, for short-term preservation. Say, okay, I want to teach my students a particular topic and we isolated organisms from the environment, and then we are going to carry out gram staining tests, capsule, flagella test, and all that. And we know that it will take us just about 2 or 3 months. We can still manage refrigeration because this is a short-term storage. But each time we do that, when we want to use the organism, we have to subculture it to show that they are still pure and they are still alive. So that is refrigeration for you. So by storing your cultures at temperatures that are, your fridge is usually typically between 2 to 8 degrees Celsius, especially in Nigeria where we don't have stable electricity. So the temperature of the fridge can vary, So usually for us around here, Our temperature, fridge temperature may be likely to be from 4 to 8 or 9 degrees. Okay, so at this temperature, the metabolic processes or activities of the organisms are, stable. It does not allow the organism to proliferate. So it slows down the activity of the organism, and then it will extend the lifespan of the organism. So what are the advantages? It is cost effective, easy to implement for short-term storage. It is used for bacteria, fungi, and yeast on agar lands or in broth. By now, you should know agar. Agar is the solid media or medium for growing organisms, and broth is the liquid medium. Maintains viability for weeks to months. But the disadvantage is limited storage time. That is, it's only for a short time. So you will keep doing, you will keep reculturing if you want to use this method for a long time. And then you stand the risk of contamination. Apart from that, you also stand the risk of losing some genetic components of the isolate. Cultures, this deep freezing is another method, the third method. And for deep freezing, the temperature is usually between minus 20 degrees centigrade to minus 80 degrees centigrade. This is not what you use an ordinary freezer for. There's usually this minus 20 degree or minus 80 degrees centigrade freezer. There are specialized freezers that keeps organisms safe. Cultures are usually mixed with cryoprotectants, for example, like glycerol or DMSO before freezing, so that the organisms will be safe, they will be viable, and they will not be contaminated. The method is also good for bacteria, fungi, and for other cell lines. The advantage is that you can store these organisms for a very long time, for years. So we have some organisms like the one I'm telling you about. It is kept under deep freezing conditions. And I have some at Naima, I have some in different places, so that if I lose, for example, some time ago in Unilag, all our cultures were dead because there was no light at the faculty for like maybe 2 or 3 weeks or a month. So another advantage is that it reduces metabolic activity. That is, the microorganisms will be stable. Their activities are altered. So you will not have proliferation, but they are preserved. The disadvantage is that it requires specialized freezers, like I said, and those freezers are not easy to come by. They're expensive. Then there's the potential loss of viability if you did not thaw it properly at the time of use. The organism has been in the freezer for like minus 20 or minus 80. The day you want to use it, you have to thaw it properly. If you don't thaw it properly, then you might lose the organism. For example, if you are so much in a hurry and you think you can just carry, your container of microorganism and put it in hot water just for it to defrost, you are going to lose the organism. So you have to allow it to thaw on the bench for a while before you can then take part of the organisms that you want to use. Another method is cryopreservation. Cryopreservation involves storing laboratory cultures at extremely low temperatures, usually in liquid nitrogen or in deep freezers, just like the deep freezing that we talk about. For this method, it prevents ice crystal formation. If you put item in ordinary freezer, you might have ice forming on that material. But for the cryopreservation, there will be no ice formation on your biological material. So the integrity of your organisms are maintained. It has the advantage of long-term storage minimal genetic changes, and it is suitable for a wide range of organisms. You can use it for a wide range of organisms. But of course, the disadvantage will be that it is expensive. It is not easy to come by. And this diagram that is taken from Franco et al. 2021 shows us the cryopreservation protocol. First, you have to sample. You have your sample, then you filter your sample, then you will apply. If you want to go for liquid sample, you apply your liquid sample, you apply your DMSO, and then you store at 4 degrees centigrade. That is ordinary freezer. Or you can concentrate your cell by filtering the water or the liquid portion away. If you don't want the liquid sample itself, you filter it, and then you apply liquid inoculum with your DMSO, and depending on the formula that is used in your lab, you apply whether it is 5% final concentration or you want to dilute it with seawater, depending on where you isolate your organism from. If you isolate it, if I isolate my organism from the lagoon or from the ocean, I know that the lagoon and the ocean is salty. So for me to have that organism to live in a natural environment where I took it from, I might mix my DMSO with 5% seawater. So the other one is freeze drying. Freeze drying. Freeze drying is also known as lyophilization. Lyophilization. Freeze drying is a dehydration process, that is, you remove the water from the cultures. And this will allow you for long-term storage at room temperature. This method is effective for bacterial and fungal cultures. No, the diagram is showing you, the diagram I've taken from Yagi et al. 2023 is showing you the lyophilizer. This cupboard here is the lyophilizer. I had the advantage of carrying out lyophilization somewhere. So you put your organisms here in its liquid form, and then switch on the lyophilizing machine, and it will keep draining or dehydrating the cultures in your tube until they all become powder. When it is ready, the machine will show sign that it is ready. It takes hours anyway. It can take up to 8 hours, 10 hours, even 12 or 24 hours, depending on the microorganisms and the quantity of liquid that you have. But at the end of the day, you have something like this in powdered form, and you can keep it for years. When you want to use this organism, they are viable. You only need to just take the quantity you need, and then you reconstitute it in your liquid medium, and you go ahead with your work. Another one is to preserve using glycerol. So you prepare your stock, like we do that a lot commonly in the lab. You have glycerol. Depends. If you use glycerol, like the organism I told you that died, I used glycerol, but that organism did not have enzyme to break down glycerol, so it starved to death. That was what happened. So for organisms, that most organisms can survive under glycerol preservation. So glycerol is like oil. So you prepare it. Most of the time we use 20% glycerol. That means you take 20 ml of glycerol and then 80 ml of distilled water, or whether it is seawater or any kind of water you want to use for your organism, depending on where you isolated them from. You mix it together, and then you take a ml of the organism, and then you put them in the stock, and you put them in the freezer, and that is it. They can spend years. So the glycerol acts as a cryoprotectant, preventing ice crystals formation and protecting the biological material during freezing. The advantage is that it is simple, cost-effective, and suitable for most bacterial cultures. Then we have the liquid nitrogen storage at minus 196 degrees centigrade. So you immerse the cultures in liquid nitrogen, and this provides extremely low temperature environments. Liquid nitrogen is very, very cold. It's very cold, but it is not a liquid. It's a nitrogen, it's a gas, so you can't see anything, but it is cold. So it can be applied to microorganisms. So it provides extremely low temperature environments and thereby prevent metabolic activity of the microbial culture, maintain cell viability for extended periods. Olaromi, I'm coming. For extended periods. Then it can also be used for viruses, bacteria, and other cell lines. The advantage is that it is ideal for long-term storage. It has minimal genetic changes, and it is versatile for various organisms. But it is very expensive and requires liquid nitrogen handling. It's a very expensive machine, and then you have to keep obtaining liquid nitrogen, which is very expensive. So Folarin, what can I do for you?
Good morning, ma. I wanted to ask a question actually. Is it
Okay.
the same liquid nitrogen they use like for bone fracture, like for injuries and stuff? Like it's always in one kind of sack bag. Is it the same thing?
It's not the same thing. Well, liquid
Okay.
nitrogen is liquid nitrogen. It depends. There are different percentages, and you can have liquid nitrogen in pouches like the one you are talking about. This one, you have them in cylinders, and it is connected to a machine, a box-like or fridge-like machines, whereby after putting your culture, you switch it on, and then the liquid nitrogen is going to be used to preserve it. You apply your liquid nitrogen. It's not only used for culture preservation. Sometimes you want to see your organism using scanning electron microscope, you have to apply liquid nitrogen. It's a whole lot of method, but this is MIC 211. This is just basic microbiology. So that is why we won't go into detail of what is beyond you. But you can read more about that. Sarah, are you ready? Yes.
Good morning, ma.
Yes,
Ma,
good morning.
which method where the organism turn
Hello?
into powder form? If you are to convert them back to the form you can use, are you mixing with ordinary water? What are you going to use to dissolve it again?
It depends on the type of organism. I said that earlier. It depends on the type of organism. For we researcher, that is why it is called a research. You do your PhD, you can do it for 10 years, you can do it for 6 years. So you have discovered the optimum temperature, you have discovered the optimum kind of substrates that the organism like. You have discovered the optimum temperature, so you just apply. Most of the time, you can use sterile distilled water. You can use sterile saline water, which is mostly used. If I know that I isolated that organism from the sea, I can take the sea water and sterilize it and then use it to dilute it. Sometimes you can even use some liquid media that will allow the microorganisms to grow. So it's just a question of taking 1 gram and then mix it together, and then you take your 1 ml and continue with your research or whatever work, wherever you are working. Did I answer your question? Sarah? Sarah, are you there? Okay, let's go ahead. Other method of preservation of cultures include silica gel desiccation. Silica gel desiccation involves drying cultures in the presence of silica gel, which removes the moisture and thereby preserving the cultures in a desiccated state. They are alive, but they are like cysts. They look as if they are not alive, but they are alive because they are in gel form. The advantages: suitable for long-term storage of fungal cultures. And then it requires minimal equipment to be able to perform. You can also store in distilled water. Some bacteria, for example, bacteria like Enterobacter or Pseudomonas can be stored in sterile distilled water at room temperature, and they will be fine because they are environmental samples. Advantages: simple and cost-effective. Disadvantages: limited to only certain species of microorganisms, because you cannot take sample from the blood or urine, maybe human sample, and then you want to store in distilled water. It will not work. But Enterobacter and Pseudomonas are environmental samples, so they can survive. So disadvantages: limited to only certain species of microorganisms, then potential viability loss over time if it stays too long, because the organisms might lack nutrients, so they might die after a while. You can also preserve using oil overlay. The microbial cultures on agar lands can be covered with sterile mineral oil to prevent dehydration. So when you apply your mineral oil, you place on top of your slants, your agar slant that contain your microbial cultures, the organisms are preserved. This is used for fungi and some certain bacterial cultures. The advantage is that it prolongs viability for months or years, and it prevents contamination, because it's not all organisms that can survive under oil. It is only organisms that have lipase that can break down oil to spoil, to proliferate. The disadvantage is that it is limited to non-motile bacteria, and it has the potential for anaerobic growth, and also for those organisms that can produce enzymes that can break down the oil. So we have mentioned the major methods of preservation, but what are the factors that can affect preservation success? You try to preserve, but sometimes you just discover that I tried to preserve this organism, but when I wanted to use it, it didn't grow again, like the one I was talking about. The name of the organism was Penibacillus. I brought all the 9 isolates and I subcultured them, and after 24 hours, I was expecting to see growth. But for that plate, for that particular organism, there was no growth. I tried it more than 5 times. There was no growth, and I knew that this organism was gone. All right, so what are the factors that may affect the success of your preservation in the lab? Microbial species, that is, some microorganisms are more resilient than the others. Some cannot, once you just take them out of their natural environment, they don't take long to die, So depending, that there are some microbial species that no matter how you treat them, I have one like that, it's called Bacillus megatherium. There's nothing, nowhere you keep it. When you go there after, this is how many years now, it will just grow. So depending on the microbial species, then it also depends on your preservation medium. The use of cryoprotectants, e.g., the use of cryoprotectants like glycerol improves the survival of your organism. The storage temperature also matters. Lower temperatures generally enhance longevity. But like I said, during your research regarding that organism, you must have studied what is the optimum temperature, what is the optimum concentration of substrates. So you should have determined, that should have given you the idea, what is the pH that these organisms like to grow in. You should have done your findings during your research. This will guide you in knowing what kind of preservation to apply to specific organisms. The handling procedures also matter. If you don't handle your organisms very well, they will be contaminated. So you need proper aseptic techniques to prevent contamination. You must have learned about aseptic techniques in the previous lectures. It also depends on your revival conditions. How did you revive? Like I said, thawing. How did you thaw it? When you brought it out from the freezer, did you just put it inside hot water to make your work faster? How did you thaw it when you brought it out from the freezer? And how did you reactivate it? So this should be done under optimal conditions. So in conclusion, for this topic, the choice of preservation technique depends on factors like the type of organisms, your intended storage duration, and what resources are available. For example, I know that lyophilization is best for my organism, but do I have a lyophilizer? I cannot say that I want to use lyophilizer when I don't have it. So it depends on the available resource. Laboratory cultures are invaluable assets in scientific research. We value organisms, we keep them, we guide them, because if you lose them, you have to go back again to start all over. So proper preservation ensures their stability and reproducibility, contributing to the advancement of knowledge across various scientific disciplines, like I said, whether it is medical, whether it is environmental, whether it is petroleum, and all that. So researchers should carefully select and employ the most suitable preservation method to guarantee the integrity and utility of laboratory cultures over time. Thank you. Any question on this topic? Like I said at the beginning, once we are done with the lectures around afternoon, I will post this slide and I'll also post the video. You will have your slide today, so you can also go back to it. Any question? Any question?
Good morning, ma'am. I have a question.
Okay.
Ma, you mentioned cultures. If we take samples from the human body, we can't use distilled water. So, what kind of liquid do they use? Is it glucose?
Like I said, depending on the organism, depending on the isolate, and depending on whatever you want to do with it. You cannot say, see, every laboratory have their own procedures for different things. Sometimes you take blood sample, urine sample, or sputum. That is a medical sample. So they have their own procedure. You can use normal saline, you can use distilled water. Normally for serial dilution, it is distilled water, sterile distilled water. But for environmental sample, most of the time, we use distilled water, or we want to use the water. If we obtain that organism from the aquatic environment, we want to take that water in that aquatic environment and sterilize it and use. So it depends. When you want to do any work, there will always be a protocol that you can use. You have a protocol, you follow the protocol to use. Yes, Nelson.
Good morning, ma.
Good morning, dear. I can hear you.
I want to ask thermophiles. What do they use for preserving thermophiles?
Thermophiles. What's the meaning of thermophile? Let me ask you that question first.
Heat-loving microorganisms.
Okay, so from all the things I've taught you today, what method do you think you will use? If you have thermophiles, will you prefer to keep them in the freezer?
No.
But you can. You can store them. What it simply means when you say thermophile is that they grow under that condition, they grow maximally. Do you understand? So when you store them, you don't want growth. You don't want them to grow. You don't want them to multiply. You want them to remain the way they are. But that thermophile now, if you want to subculture it when you want to use it, you have to use the right temperature, which is that high temperature. You cannot want to culture a thermophile using 27 degrees centigrade. It will not grow. Human culture will grow at 37 degrees centigrade. So if you are talking about thermophile, maybe it's 40 degrees, maybe it's 45 degrees. And sometimes all these organisms, even though their optimum temperature may be 45 and above, that does not mean that they cannot still grow at other temperature. It's just that they will not grow optimally. It's just like, for example, a child that is malnourished. The child is growing, but the child can have a stunted growth. I'm just using that example. Do you understand what I've said?
Yes, ma. Yes, ma.
All right.
Thank you.
Thank you. So, any other question? So, should I give you a minute to take a breath so that we can continue with the other lesson? Like I told Salako, yes.
Ma, so I have a question, ma. Ma, this particular machine, this lyophilizer that you said carries out lyophilization. Ma,
There's noise
what
at
kind
your
of
end. There's too much vibration. I can't hear you.
Sorry, ma, I mean that lyophilizer machine, ma.
Can you do what?
That lyophilizer machine, what industry do they use that machine, ma? Or
Any laboratory, the laboratory where I did my PhD in the US, they have a lot of lyophilizer. Where I did my postdoc in Germany, they have a lot. They have these things as if you just have radio or television in your house. But it's not the same thing with us in this part of Africa. We don't have it. Most labs do not have it. Maybe some private lab will have it. Or maybe places like NIMA. I'm not even sure NIMA also have it because they may not use it. They may not be using it. So most of them in this part of the world, we use freezer or -20, -80. I've not been able to use any lyophilizer over here or around here. All right. So in the absence of no question, we have taken a deep
Sorry, hello ma. Good morning, ma.
Thank you. Go ahead.
Ma, please, ma, I don't think I understand the case of that thermophiles that you explained earlier.
Okay.
Can you please explain better, ma?
What do you mean by thermophile? I'm asking you, what is thermophile? Who is that person that asked question? What is thermophile?
Yes, ma. You said I didn't really get the definition that you gave, but you said they grow maximally.
I did not give you the definition, and we're not here to talk about thermophiles. We're here to talk about storage, right? Preservation.
Yes.
So whether it's a thermophile or mesophile, different organisms, you can use different method depending on your specific organism. So if you have a thermophile and you want to store it using glycerol overlay or oil overlay, you can use glycerol, you can use depending on the organism. So you can store them in glycerol, you can use overlay, you can store them at deep freezing, like -80, but when you want to grow them, when you want to start working with them, you have to use the appropriate temperature, otherwise they will not grow and you will think that something is wrong. If an organism grows optimally, at 47 degrees centigrade, and you now put it in the incubator at 27 degrees centigrade, you will not have the optimum growth. That is what I'm saying. For preservation, it is not a big deal, because during preservation, you want their metabolic activities to cease. But you will ensure that the organisms you are keeping, that the method you are using to store it will not affect the integrity or viability of that organisms later. So if I am to store a thermophile, for example, I would rather use methods like oil overlay or glycerol rather than freezing. If you isolate anaerobes from human, maybe from otitis media in the hospital, the distance between the hospital and your laboratory may be up to 1 or 2 hours. If you isolate that anaerobe before you get to the lab, if they are facultative, if they are anaerobes, they would have died. So carrying them from the hospital to the lab, you have to preserve them in what we call charcoal agar or amine to preserve their longevity, so that you take it to the lab and then you quickly culture it under anaerobic condition. But if they are facultative anaerobe, that means they can still survive some level of oxygen. So 1 or 2 hours may not make a lot of difference, but you have to make sure that within those few hours, you quickly culture them using anaerobic conditions. So it depends. That was why I said it depends on the organisms you are dealing with. Any other question? Because I want to move to the next topic. Any other question? C, you are going to have CBT. And this CBT, you have questions, mixture of objective. So different people will have different questions. And because I'm not here to teach you about under this preservation, of course, you will see other things like cycrophiles, mesophile, So, but you have to pay attention to detail. Just be calm. Every question that will be asked will come from your slides. It will not be taken from any other place. Your slide is where your questions are taken from, and your questions are ready. So now we want to move to another topic. All right, quickly, we want Polari.
No, I just wanted to ask something. Like, is the class also streamed on YouTube? Because some people are saying they can't join again because of.
I did, except if something is wrong, maybe something is not working. That was what I did first, even before joining this.
Ma, please, can you resend the YouTube link so others can join? Like, the class is filled up already.
Well, I'm not sure if I can do that now. Just let them know that I'll post this slide immediately, and then I'll try to send the link. I'm not sure what I did not do right, because by now they should have seen it. So, what did I touch? I have touched something. All right. Let's quickly look at microorganisms in relation to food. Microorganisms in relation to food. That is, what role do microorganisms play in food, in relation to food? You must have done microorganisms in relation to medicals. So at the end of this session, you should be able to summarize the history of microorganisms in food, which you have learned under history. You must have learned some part of it under the history of microbiology. You should be able to identify the category of microorganisms found in food. You should be able to identify the primary source of microorganisms in food. And you should be able to highlight the beneficial and harmful roles that microorganisms play in food. So microorganisms play significant role in food, affecting whether it is food quality or the preservation and safety. You preserve your food, but sometimes you just go there, you say that this food is spoiled. Sometimes you keep your food in the fridge, your cooked food in the fridge, and after a week, you see some whitish things growing on it, Microorganisms have spoiled it. So microorganisms affect your food quality, the preservation of your food, and the safety, because consuming spoiled food might lead to sickness. So microorganisms are beneficial. It is not all microorganisms that are harmful. As a matter of fact, beneficial microorganisms outweigh the pathogenic or dangerous organisms. So some of them are beneficial because they aid in production of food, like in fermentation, and in digestion of your food because you have them in the gut. There are microorganisms right from your mouth, in your saliva, that helps to release enzymes to digest your food. Others cause food spoilage and foodborne diseases. So understanding the interactions of microorganisms with food is a necessity for food safety, for biotechnology, and for public health. So this is what we want to look at briefly in the next 1 hour. All right. This history of microorganisms in food is a very long one, even before the scientific era. Before we begin to say, okay, we now have scientific era, before or the pre-scientific era, we already have microorganisms as relates to food. Long before microorganisms were known by man, when man do not even know that microorganisms are existing, we've been applying microbiology or microorganisms. For example, I know that my grandma does ogi. And when I was very young, I did not know what she does. But I know for sure one thing: once the ogi is getting finished, she does not allow it to finish. She buys another corn or guinea corn and soak it. Then she will scoop a handful from the initial ogi before it finishes and put it on the corn that she's soaking. At that time, I used to wonder, what is this woman doing? But when I now got to 400 level and learned food microbiology, I noticed that scooping from the initial ogi that is already fermented, putting that in the new corn or guinea corn is only helping to release microorganisms responsible for fermentation and their enzyme into the new guinea corn or the corn, so that the enzyme will quickly act on it and hasten the softness. So, and I practically practice it. I soak some corn without putting old ogi and another one with a handful of ogi. And I discovered that the one that have the previous ogi, in 2 days is already soft. But the one without the old ogi was still hard and took about 5 to 7 days before it became softened. So our forefathers were practicing they were using microorganisms without them knowing. They do know that their meat can get spoiled, but they know that when they dry this meat, it will be preserved. When they dry or smoke their fish, it will be preserved. They did not know that by drying or by smoking, that they are removing the water. And when they remove the water, microorganisms will not be able to survive. They did not know. So they use this method of fermentation to do local ogi, iru, or kito that is mostly drunk in the north and central area like Benue. But they did not know they were practicing it. But we also have some evidences that people have been practicing food microorganisms, practicing food microbiology, and they've been using microorganisms in food unintentionally. For example, the Babylonian, as far back as 7,000 BC before Christ, the ancient Babylonians have been producing beer. The Sumerians, for example, around the 3000 BC, they are livestock breeders. And because they are livestock breeders, they are also dairymen. So they make use of a lot of dairy products like yogurts. Even in my own local environment where I came from, they didn't know anything about science or microorganisms, but they do produce cheese from cow milk. They milk the cow, and then they produce cheese from it by adding one or two things. So butter, salted meat, fish, fats, dry skins are all known to be associated with the cultures of the Sumerians, even though at that time they are not aware about microorganisms. So milk, butter, cheese were also produced by Egyptians as early as 3000 BC. That is on the advantageous side, on the beneficial role of microorganisms. On the other hand, the undesirable role of microorganisms has been dated far between the birth of Christ and after the death of Christ, like 1,100 years. For example, there was a case of ergot poisoning that killed quite a lot of people. I think in France, yes. People were dying, but they did not know why they were dying. But later they discovered that it was ergot poisoning. They call it ergot poisoning. But later they discovered that ergot poisoning was caused by injection of alkaloids produced by a fungus known as Claviceps purpurea. So this organism, maybe when there was humidity, grow on grains and caused many deaths during the Middle Ages. Over 40,000 people died of this ergot poisoning in France. But it was not known at that time that it was caused by a fungus. People could not link meat spoilage with microorganisms at that time. But they just know that they put anything on the table, it gets spoiled. But if they add salt or dry it or boil it, then it takes time before it spoils. But they were not aware that they were dealing with microorganisms. Kepler was the first scientist to suggest the role of microorganisms in food spoilage in 1658. Before other scientists like Spallanzani and Swan demonstrated the idea of the heat preservation of foods. Canning of foods had its beginning in 1795 by Francois Nicolas Appert, who first preserved meat in glass bottles. He discovered that if he puts meat in glass bottles that have curved neck so that air cannot go in, if he boils the meat very well, that the meat will not spoil. But if air is allowed to go in, the meat will spoil. Pasteur was the first to understand the role of microorganisms in food. In 1837, what did he do? He showed that it was microorganisms that caused souring of milk. And in 1860, he showed that heat could destroy undesirable organisms, both in wine, in beer, and in milk. This is why his method is called pasteurization. So if you want to know more about the history of microorganisms in food space, you can read more about it yourself. So the types of microorganisms that is associated with plants, products, and food in their natural state can help us to predict the general type of microorganisms to be expected in the food product at a later stage. Because our food is either of plant origin or animal origin. So when you have a fresh food and you isolate the organisms you see there, maybe the natural organisms that are supposed to be there. But later when you start seeing changes in the colony of microorganisms, you know that these food products have been contaminated. So the safety of a food is based on what is the total number per gram of microorganism or per meal. If I have a cup of juice, or if I have a bottle of juice or bottle of water, and I try to isolate microorganism, aseptically without contamination, I will see the total number of microorganisms I have there. If I now put it in a cup and leave it on the bench for 6 hours and I isolate again, the total number of organisms I will see per meal will be more, because it has been exposed and it has been contaminated. And this will also show that different types of microorganisms are now present in their number in that product. So what are the factors that influences the presence of microorganisms in food? What are these factors that influences the presence of microorganisms in food? So the general environment where you are can influence the presence of microorganisms. Please mute. There was a study myself and my colleague did about 4 or 5 years ago. We noticed that in Lagos, there are vendors that are, selling food. They carry it at Oshodi, Ikeja bus stop, or Jota bus stop, or any bus stop. They also have salads. If you notice, they put their coleslaw or salad in one whitish plastic without ice. They hawk it. So we compare that hawked salad in that Oshodi area that is very rough with a lot of people. They are selling the food, they expose the food. Some people are talking, saliva and everything. Some people are sweating, some people are fighting. And then compare it with coleslaw or salad or rice that is maybe sold in a restaurant at Ikoyi. We did all that and discovered that the environment will affect the presence of microorganisms. The microbiological quality of the food in its raw state or processed state is also a factor that may influence the presence of microorganisms in your food. What I'm trying to say is that raw food, for example, if I have a garden of vegetable, vegetable, when you want to eat vegetable, you want to make it half cooked so that you don't lose the nutrient. But if I have a garden well fenced, or I use a screen house, you discover that rodents cannot go in there. People cannot urinate there. They cannot there. So even if I pluck that vegetable and simply rinse it and eat, I will not have problem. Compare that with somebody who planted vegetable at the back of their house in a very rough place. People will wash their fish and meat, raw water, they will pour it there. Small children will go there and just urinate. Some people will pass excreta and bury it there. By doing that, maybe some people are infected with some certain organism. By passing stool, they have released some dangerous organism. If I pluck such vegetable and I don't wash it thoroughly with salt and allow it to cook properly, the person will be infected. So rather than having nutrients, the person will be sick. So the microbiological quality of food in its raw state also matters. We influence the presence of microorganisms in the food. Then the sanitary condition under which the food product is handled and processed. You go to Red Block or wherever you eat. Did you bother to look at the back of the restaurant? Do they urinate there? Do they wee there? What activities goes on around where you eat? So the sanitary condition under which the food is prepared also matter. The adequacies of subsequent packaging, handling, and storage conditions in maintaining the flora, that is the flora of microorganisms at low level. You finish cooking, but you just left it on the bench instead of putting it in the fridge. After 24 hours, the quality of the food will be compromised. Or you cook fresh food and then you take a dirty plate and you pour your food. Then the food will be affected. Some people leave their dirty plates after eating. They will not wash it until after 2 or 3 days when they finish using all the plates. And by then, some plates already growing black. Maybe they ate a bar or something. You know, the plate is already growing black or greenish something. They simply take their sponge and just wash it. Whereas the fungi that is already grown on the plate might still be there, though you are not seeing it. So putting your food there is a risk. So here on this table, we have the list of some microorganisms that you can find in your food. We have the set of bacteria, the molds. You know, when you say fungi, fungi consist of molds and yeast. You must have done that at the beginning of this lecture, so you know the difference between molds and yeast. And then you have the protozoans, which are mostly parasites. So for bacteria in food, you can find Acinetobacter, Enterobacter, which may be from the environment, Escherichia coli, which may be from the food handler. Bacillus campylobacter may also be from the food handler. Maybe somebody is picking, you are cooking food, or you are cutting meat or preparing salad, and that is when you are using your hand to scratch your head or scratch your skin or picking your nose, or you just go to toilet, you did not wash your hand with soap, and then you go back to that salad, you are introducing some microorganisms. You can also have Clostridium, Listeria, Salmonella, and all that. Then for molds, you can have Aspergillus, Botrytis, Cladosporium, Fusarium, Mucor, Penicillium, Rhizopus that spoils bread. Then for the yeast, you can have Candida. Most of the time, Candida Saccharomyces are very useful, and the Brettanomyces, they are useful. Yeast are generally useful, though you have some also that can cause diseases. You have some species of Candida that can cause diseases, but most of the yeasts are very useful in the industry for fermentation. Then, of course, you have protozoans like Cryptosporium parvum, Entamoeba histolytica giardia, which you can find in your water, and Toxoplasma gondii. Most of these are parasites, and they can cause diseases. So what are the primary sources of microorganisms found in food? Soil and water is one, because many of the bacteria and fungi that inhabit both soil and water share a lot in common. Soil microorganisms enter into the atmosphere by the action of wind. You have organisms in the soil. When there's wind, they can be taken into the air, then they settle on whatever they want to settle on later. The same thing when it rains. Air microorganisms can descend and fall on surfaces of the soil or table or wherever. And when you use your hand to touch them and handle your food without washing, you can introduce them. The aquatic microorganisms can be deposited onto soils through the action of cloud formation and rainfall. Some aquatic organisms are unable to persist in soil, especially those that are indigenous to marine water, like Alternoromonas. Those are environmental species. So they require salinity for growth, so they may not be able to persist in soil. So these organisms can be from soil where you grow your food, like vegetable, or from the water that you use for irrigation. If you use water, maybe from drainages, you can imagine people urinate, people bath, people can wash their bum. So what do you expect on that vegetable? So your soil and water is a primary source of microorganisms that you can find in your food. The plants and the plant products, the microorganisms that persist on plant products usually do so because they have the capacity to adhere to plant surfaces. If not, when rain falls, they would have fallen off. And because they also obtain their nutrients from such a plant, they stick to the plant. So if you want to eat salad, for example, people washes it with maybe salt water or with vinegar so that they can dislodge or detach the microorganisms from such plant products. Food utensils, like I said earlier, as more and more vegetables are placed in some containers, a normalization of microorganism flora will occur. Also, in the house, we cut fish, we cut our meat, we have cutting block or what we call the chopping board. Some houses, they don't have separate cutting block or chopping board for meat and fish. When they want to eat salad, they use the same chopping board. They want to cut fish and meat, they use the same chopping board. But the truth is, when you cut raw meat or raw fish or raw chicken, the microorganisms might be transferred to your vegetable or salad. You cannot say because you have used sponge and soap to wash the cutting block or your chopping board that it is sterile. It is not sterile. So ideally, you should have different cutting board for salad, that is for ready to eat food that you don't need to sterilize, and another one for raw food like
Just know that you are told another person that money is not the main thing.
Can you please mute yourself? You are disrupting this lecture, please. The same thing with your grinder. If you use grinder to grind pepper, to grind egusi, and then you want to take your smoothie, your vegetable, and you use the same grinder without properly washing or sterilizing, you might get infected through that process. So the cutting boards, the grinders are contaminated from initial samples. You know, when you are tired, at times you grind your egusi or pepper, you just carry the blender, you just rinse it and just leave it like that instead of washing it thoroughly with water and soap. So this leads to a buildup of organisms. Intestinal tract of humans and animals is another source of microorganisms in food. The intestinal flora, that is intestinal microorganisms, consists of many microorganisms. Our intestine consists of many microorganisms. Among them are pathogenic organisms like Salmonella, Enterobacter, which may be expected in physical waste when the person passes feces, along with intestinal pathogens, including protozoan species, like somebody that is stooling and vomiting. Then you just, after using the toilet, you just got up, you just rinse your hand, you did not use soap and water. Then you went ahead to prepare food for your family. The next thing is that everybody will start stooling and vomiting because you have ended up transferring the salmonella or the organism that is responsible for vomiting and stooling. So the intestine of human being and animal can also introduce microorganisms into food. That is why sometimes they will tell you you have to cook your meat very well. You have to cook, because there are some organisms that can affect animals that can also affect human beings. Then the food handler is another one. I've explained that over and over again. The way you handle your food will determine the type of microorganisms that you will find in your food. So scratching your hair, picking your nose, talking during cooking, especially or during serving, when you are serving, maybe ready to eat food, when you talk, you introduce microorganisms to it. Your poor personal hygiene practices generally, introduces microorganisms into food. So food handlers are another source of introduction of microorganisms into food. Animal feeds. The animal feed can introduce organisms. For example, salmonella can be introduced to poultry, to your chicken and other birds and other farm animals. It is known that Listeria monocyte, that dangerous organisms in dairy and meat, is transferred through silage, that is the food, the fodder that is packaged in silos. When they package, over here, we give our animals either the dry grass or green grass to eat. But in the Western world, they usually have a silos where they keep their green fodder. If it is not properly dried and kept, it can be infected. So by the time the organism, the animals or birds take this food, they get infected. If human beings consume these animals without proper cooking, then the human being also gets infected. So people that eat spoon, people that eat half-cooked meat, half-cooked fish can be affected with such microorganism. That is why some people will go to party and 6 hours or 8 hours later, they start vomiting. It is because they have ingested microorganisms along with their food. I'm not enjoying this class because some people just refuse to mute. When you talk, you make it difficult for your lecturer because the lecturer now have to be talking, on top of her voice. Animal hides, the type of microorganisms found in raw milk. You can get your raw milk from your cow. Real one, please, are you in the class?
Yes, ma'am.
Please, can you control that person?
Okay, ma'am.
So, because I can't start looking at over 300 persons here to know who is talking. Is this the first time you are attending classes? I'm the last lecturer. By now, you should be used to the rule of virtual lectures.
Ahmed de Comfort, please mute your mic.
So the animal hides, the type of microorganisms found in raw milk can be a reflection of the flora under the udder of the animal. The cow that gives milk have breasts called udder. So if that cow have skin infection and the nipple of that cow is infected, the milk that will be gotten out of that, oh my God. Further disturbance means I will stop this lecture and simply send the slide to you guys to go and read, and that is it. If you cannot control your colleagues, I will send the slide and I'm done with this lecture. So the udder or the nipple of the cow or the animal, when infected, can affect the milk that will be obtained from that cow. So usually the teeth or the nipple of the cow should be cleaned before the milk is obtained. And then the person that is milking the cow should have gloves or the hands should be properly washed. Otherwise, if you are using a hand that is dirty to milk the animal, you might even be the one introducing contaminants to the teeth of the animal and to the milk. And then your storage container, for example, you are milking, the milk container should be sterilized, should be sterile before you milk. Then you can also have organisms from hair and dust, like I explained earlier. So microbes in food. You have different types of foods, but generally foods are composed of carbohydrates, protein, or fat and oil. So all of these serve as substrate, that is media, food for different kinds of organisms. Like I told you, you cannot see microorganisms with your naked eye until you grow them in a media. We call it media, but it is food. It is food for the microorganism that is called media. You either call them media or substrates. So because of the presence of different enzymes, microorganisms can break down food components. So those organisms that have the capacity to break down carbohydrates will break it down. For example, organisms that have cellulase enzyme will break down cellulose. Those that have lactase will break down lactose. The same thing, protein. You have organisms that prefer protein. You have organisms that have lipase that can break down fat and oil. So because of the presence of different enzymes, microorganisms can act on food, producing or releasing different products. For those that are microbiology students, when you get further in your 400 level and masters, you will determine how you can use microorganisms to ferment and then get different products from that. So carbohydrate foods, for example, cereal, are acted on by the carbohydrate fermenting organisms, producing what will be the end product, acid or alcohol or gases. And I just mentioned an example. Proteinous food like meat or fish are acted upon by proteolytic microorganisms, resulting in production of amino acid amine, ammonia, or hydrogen sulfide. So when I talk about acid, alcohol, and gases being produced when organisms act on carbohydrate food, acid, alcohol, and gases are the end product. Then when they act on proteinous food like meat or fish or egg, the end product will be either amino acids, which are the building block of protein. They are simply breaking it down. Amino acid or amine or ammonia or hydrogen sulfide that smells like rotten egg. And for the fatty acids like oil or butter, when they are acted upon by lipolytic microorganisms, that is organisms that can produce lipase enzyme, the end products are usually fatty acids and glycerol. So you can find out as an assignment, you are not submitting, find out 4 examples of microorganisms, each that can act on carbohydrate, protein, and fatty acid, and the enzyme that they produce. I gave you an example each earlier. Classification of food. We want to classify food because the classes of food will also determine how quickly they get spoiled. So the physical or chemical characteristics of food determine their degree of susceptibility to microbial activity. So on the basis of stability to microbial attack, or according to the speed with which food can spoil, food can be classified into 3 types. You have the perishable foods, the semi-perishable foods, and the stable or non-perishable foods. For the perishable foods, these are foods that spoil quickly, like meats, fish, poultry, milk, egg, and other dairy products. Vegetables and fruits among the perishable food. Even though we have fruits among the perishable food, we have the fairly semi-perishable fruits like apple, pear, or nut. They are semi-perishable. If I put an apple on a table, it can stay there for like maybe a week, but I cannot put my watermelon, banana, or pineapple for that long. So some food are perishable because they spoil quickly. Some are semi-perishable because they spoil less quickly. Then we have the stable or non-perishable food, those that do not spoil when you keep them under normal storage condition. I'm saying under normal condition because if you have rice, flour, or sugar, they do not spoil. They are non-perishable food, but that is when you keep them under the normal condition. If, for example, you keep cereal or flour or sugar in an humid environment where you have heat and humidity, the flour or the cereal will absorb some quantity of water. So you can have microorganisms, especially fungi, that will begin to grow because fungi do not require too much water to grow. They only require mist, a little amount of water to grow. So food samples that spoils quickly, you discover that they have water. And water is very necessary for microbial growth, especially bacteria. So the less water a food sample have, the more stable they are. So microorganisms in food can be classified into 3. You have beneficial microorganisms, those organisms that are used in food production and fermentation. You have spoilage microorganisms, those that cause deterioration in food quality. They deteriorate food. Sometimes you have food and then the next day it's smelling. You can smell it that it is getting sour. So the food will be deteriorating. Those are being deteriorated by spoilage microorganisms. Then you have pathogenic microorganisms, those microorganisms in food that can cause foodborne diseases. So let's look at the beneficial microorganisms in food. These microorganisms are exploited. They are used in the food industries to produce desirable food products and to enhance the quality of food. For example, the lactic acid bacteria that is commonly called LAB They are used in the production of yogurt, cheese, and other fermented dairy products. So an example of such lab is lactobacillus, streptococcus, and leuconostoc. And for the yeast, you can use them in baking, brewing of wine or beer. And then we have examples like Saccharomyces cerevisiae. Then molds can be used in cheese production, e.g. penicillium. So on this table, I have beneficial microorganisms in food. And I'll just tell you the products, the microorganisms that produce them, and the role they play in such food. So we have beer production. The microorganisms that are used in beer production include Saccharomyces cerevisiae, Saccharomyces casbagensis, and Saccharomyces ovarium. What role do they play in converting cereals to beer? They convert the sugar in cereals to alcohol and carbon dioxide. I already said that earlier. So they produce changes in protein and other components that modifies the flavor of the beer. That is why this flavor differs from one industry to another, and there's a kind of competition. For example, look at Guinness and NBL, Nigerian Breweries Limited. There's serious competition. You discover that one produces lager beer. What is the other products that looks like lager beer for the Guinness? I've forgotten the name. I can't remember the name, but you discover that the lager beer of NBL is still the best, and Guinness have not been able to meet up. But when it comes to the black liquor, that is stout, the legend that is produced by NBL cannot beat stout that is produced by Guinness. People that drinks this alcohol, they are the only one that will tell you that, oh, they like the flavor of Guinness stout, but they like the flavor of NBL lager beer. Every industry have their own secret, the microorganism species they use. They have their strain and they guide it jealously. So hardly will you see a brewer in NBL moving to Guinness, because they will keep this microbiologist jealously, because the microbiologist can actually take this strain to other industry if they don't treat him well. And then those industries will be able to compete successfully. Look at palm wine, excuse me. So take a look at palm wine on this table. Palm wine have mixed flora of microorganisms, but we have isolated microorganisms from fresh palm wine before. So you can have Saccharomyces cerevisiae that has been isolated in fresh palm wine, Candida species, Schizosaccharomyces pombe, Lactobacillus species, and all that. But these organisms can ferment the palm wine, and after fermentation, they may become acids. Cheese is another product. The microorganism that is used to produce cheese includes Brevibacterium linings, Penicillium roqueforti, Streptococcus salivarius, subspecies Thermophilus. This species is used for the ripening of cheese. And then wine. Wine can be fermented with Saccharomyces ellipsoides of different strains. They can also use Saccharomyces ovarius and Saccharomyces oviformis. What role does this microorganism play in the fermentation of wine? Convert sugars to alcohol and also modify flavor. We also have some other beneficial roles of microorganisms in food. For example, fermented meat products like sausage, liver spreads. It is the job of microorganisms that helps in the fermentation. Fermented dairy products like yogurt, buttermilk, sour cream, fermentation of foods like ogi, iru, and dada, and the local drinks. The microorganisms are also used as food supplements and additives. For example, the single-cell protein and edible mushrooms. Single-cell protein and edible mushroom is known to be consumed, for the protection of the eyes or treatment of some eye diseases. We also have bacteriocin that can be obtained from some bacteria. Bacteriocin is a bacterial metabolite. It's a metabolite that is released from the activity or metabolic activity of bacterial species, for example, bacillus, and can be used as food preservatives. Lactic acid bacteria isolated from traditional fermented food like iru or ogi have health-promoting benefits. Fermented food have health-promoting benefits. They are also known collectively as probiotics, which is good for human health and body protection. In the digestion of food, these organisms ward off invasive organisms. For example, Bacillus subspecies ST13, after safety study, it has been reported to show antagonistic activity against enteric pathogens like Staphylococcus aureus or Salmonella typhimurium. That is, if Bacillus subspecies ST13 is included in your yogurt, for example, and you drink it, if you mistakenly take contaminated food that contains Staphylococcus aureus or Salmonella, the Bacillus species will fight this Staphylococcus and will not allow it to be able to survive. So that is why probiotics food are good. So you now have live yogurts. You have yogurt that have live organisms with them to protect you. They are all over the supermarkets. Microorganisms help to improve animal feed for ruminants and non-ruminant animals, e.g., white rot fungi. Fermentation of wheat straw has enhanced the protein content and improved the digestibility of different plants for for animals to be able to digest it easily. So let's look at the spoilage microorganisms. Spoilage microorganisms in food. Spoilage microorganisms cause food deterioration, leading to undesirable changes in taste, texture, color, and smell. If you keep your plate of food on the bench or in the kitchen till 2 days or in the fridge for a week, what do you do? What is the normal thing we do first? We remove it. We try to smell it. Is it smelling funny? Like the smell have changed? Then, if the smell changes a bit or the smell is still intact, what do you want to do? You want to use a spoon to pick some of the food, use your hand to feel it. If it is drawing, you say, oh, this food is spoiled, it's already slimy, it is spoiled. You, even without going to school, our grandmother knows that when the food is slimy, that it is already spoiled. This is because there are organisms there already. They've carried out their activity and they have released some metabolic products, so you can feel the slime. So some people still want to go ahead to eat such food. So what do? You have to heat it properly. By heating it properly, you want to kill the organisms before eating the food. So even though the taste and the smell have been altered, some people will still eat the egusi soup that is smelling of taste. But if it is not properly heated, then the person might start vomiting. Then it becomes a case of penny wise, pound foolish, because you don't want to throw away 3 spoons of soup. The person might end up in the hospital spending 300,000 instead of just throwing that soup away. So you have bacteria, for example, Pseudomonas, Bacillus, Clostridium, Lactobacillus, that can spoil dairy products. Then fungi, you have Aspergillus, Rhizopus that can spoil bread, Penicillium, they cause mold growth in bread and in fruits. Then yeast can cause fermentation in fruit juices. You have your fruit juice or your smoothies and you want to enjoy it, but maybe NEPA decide to show you and you just discover that 2 or 3 days later, your juice is already fermenting and the taste is off. So it has spoiled. The milk and milk products are also spoiled by lactic acid bacteria. So the growth of molds can spoil carbohydrate-rich food like bread, jam, and some other fruits. Proteins such as meat can be spoiled by anaerobic breakdown also. It causes putrefaction under anaerobic condition, yielding foul smelling in mind, like rotting. When you start smelling that something is rotting. Then butter, even though you think that butter, you can keep it anywhere, anyhow, but after a while, there can be rancidity of butter. There will be a bit of off taste, and this is caused by lipase-producing microorganisms. The organisms that can produce lipase, an enzyme that can break down the butter or the oil. So the pathogenic microorganisms in food, these microorganisms pose serious health risks when consumed in contaminated food or water. When we talk about food, we are talking about water, drinks, and anything that goes into the mouth. Bacteria like Salmonella, Escherichia coli, Listeria monocytogenes, Clostridium botulinum, Staphylococcus aureus, they cause foodborne diseases. For example, cholera is caused by Vibrio cholerae, and dysentery is caused by Shigella dysenteriae. Viruses like norovirus, hepatitis A virus can be taken with food or water. And hepatitis damages the liver, because the liver is responsible for the elimination of everything we take in, right? With food, drugs, and whatever we take in is actually filtered out by the liver. So if the liver is sick with hepatitis, it is not far-fetched that the person is going gradually. Then we have parasites like Giardia lambda, Toxoplasma gondii, Cryptosporium that can be ingested with water. So let's look at the microbial contamination of food and sources of contamination, which we have looked at before anyway. So food can become contaminated with microorganisms at different stages. Primary contamination from soil, water, and raw material. Those are primary contamination. For example, the soil where people are urinating and defecating. If you decide to plant your lettuce and you cannot cook lettuce, so it's likely that that lettuce is already contaminated. Secondary contamination is through handling, processing, and packaging. I've explained that all earlier. How you handle the food with your dirty hand. You pick your nose, you scratch your head, you scratch your skin, you put your hand on the railing of staircases without washing, and you just go straight and begin to cook and preparing the food. Then cross-contamination can also occur due to contact between contaminated and uncontaminated food. So we have already mentioned several sources of contamination, which include soil and water. Raw foods like vegetable and seafood can carry microbes. And most of these things are foods that are not likely to be properly cooked. So we have to ensure that even though vegetables cannot be properly cooked because you want to retain the nutrients, but you must know how to sterilize them, either by first soaking them in water, in salt water, or using vinegar water to rinse them. Seafoods have to be properly cooked. Then the food handlers, poor hygiene can introduce pathogens. I've explained that earlier. The processing equipment like the chopping board, your grinder. Also, poorly sanitized equipment can harbor bacteria. Storage condition, improper temperature and humidity can promote microbial growth. I explained that of humidity using grain. You say, okay, your grain cannot spoil, it is non-perishable, but you just carry it and put it on the floor in your house. Meanwhile, maybe you live around the waterlogged area. Then after some weeks, you discover that the rice or the grain is caking, and then they begin to change color. What are the factors affecting microbial growth in food? What are the factors affecting microbial growth in food? Microbial growth in food is influenced by intrinsic and extrinsic factors. Intrinsic factors mean the composition of the food itself, the inherent factor of the food. For example, the pH of the food. You remember pH, alkalinity, salinity. Remember that your ruler, 0 to 14. 0 to 7 is 0 to 6 is acidic, 7 is neutral, then 8 to 14 is alkaline. So different food have different pH. So acid food like citrus fruits can inhibit bacterial growth because some bacteria cannot withstand acidic condition. Most of the time, bacteria prefer neutral condition for them to be able to grow. So when the food type is acidic in nature, it will take time for it to get spoiled because it will inhibit some bacterial growth. Moisture content, which is related to water activity. Dry food inhibit microbial growth, and I've explained that earlier. Bacteria, for example, likes water. Fungi do not require much water as bacteria. So with little moisture, fungi will grow. Nutrient composition. Protein-rich food support bacterial growth. Even human beings know protein is very rich. So bacteria also love proteinous food. So nutrient composition is a factor that will affect the growth of microorganisms in food. So that is why food like fish, meat, eggs, poultry products, chicken, they get spoiled easily because they are nutritious. And so bacteria also enjoy these nutrients. Another factor is oxidation reduction potential. Oxidation reduction protection of its substrate is the tendency with which the substrate loses or donates electrons. Substrates that loses electrons will spoil easily because they can disintegrate easily. Biological structure is another one. When you have eggs that have its egg shell intact, they can be in your kitchen for 2 weeks, 3 weeks. You know, we don't use the expiry date in Nigeria, so it can be there as long as when you break it, you still see the egg yolk standing. You say the egg is good. The egg shell is protecting it. But if you have the egg, the shell of that egg cracked from where you are bringing it from, it does not stay long to get spoiled. The same thing with fruits. You know, the fruits have covers. The peels keep them. But when the fruits already have holes or the skin is broken, that fruit will easily get spoiled. As a matter of fact, when you pick an apple or orange or any fruit at all, and you begin to see hole or damages, the best thing is just to leave it because it is compromised already. So biological structure of a particular food helps in delaying spoilage. Then we also have some food types that have antimicrobial constituents. For example, cloves. Cloves have eugenol, and eugenol does not easily support the growth of microorganisms. Garlic also have allicin. Allicin does not easily support the growth of microorganisms. That is why you see you keep cloves, you keep garlic, you keep ginger on the bench for months, and they are just there because they do not easily support the growth of microorganisms. So let's look at the extrinsic factor. We just finished talking about the intrinsic factor, that is the food composition itself, the factors of the food itself. So let's look at the environmental conditions, which are the extrinsic factors of the food. The temperature where the food is stored. For example, psychrophiles, even though you store your food in the refrigerator, you wonder why after 5 or 6 days, the food is still getting spoiled. It is because you have organisms that are called psychrophiles They are cold-loving organisms and they can grow at refrigeration temperatures. The mesophiles, that is moderate temperatures, like room temperature, include many pathogens. So if your food is just kept on the table, it gets spoiled easily. Thermophiles, heat-loving organisms, grow at a high temperature. So if you keep them on the bench, they might survive for long until the heat favors them. Then you have the psychrophiles that grow well between -5 and 15 degrees centigrade, but they have their optimum growth between 12 and 15. I was talking about optimum growth, and I was talking about many organisms can grow at varying temperature, a wide range of temperature. But when you want them to grow optimally, there is what is called optimum temperature. So psychrophiles that are cold lovers, they can grow well at the temperature range of -5 and 15 degrees centigrade. But they will grow optimally between 12 to 15 degrees centigrade.
I don't understand.
The psychrophiles grow well at or below 7 degrees centigrade, but they have the optimum temperatures between 20 to 30 degrees centigrade. The mesophiles grow between 20 and 45 degrees centigrade, but the optimum temperature is between 30 and 40 degrees centigrade. Whereas the thermophiles can grow well above 45 degrees centigrade, but their optimum temperature is usually between 55 and 65 degrees centigrade. Mesophilic and psychotropic organisms cause more spoilage of food than other type of organisms because of the condition. Mesophilic condition is common. It's just like room temperature. So mesophiles with an optimal temperature of 37 degrees centigrade, often of human origin. Real one. Is your class captain? Can you tell your colleague?
This person should mute his mic.
What kind of?
She's not from microbiology.
Mute your mic.
So, I said in a well.
Afolabi, mute your mic.
Ma, please, can you just mute everybody?
Mesophiles with an optimal temperature of 37 degrees centigrade are often of human origin and include foodborne pathogens such as Salmonella, Clostridium perfringens, and Staphylococcus aureus. Another environmental condition is oxygen requirements. For example, aerobes, those organisms that require oxygen, e.g. Pseudomonas, if they don't have oxygen, they cannot survive. So if you can the food or bottle the food, it's not likely you are able to obtain Pseudomonas. Then we have the anaerobes. Anaerobes often affect our bottled or canned food because anaerobes can survive under anaerobic conditions. So you may think that you have a canned sardine or bottled juice, packaged juice without oxygen, but they can survive if during the production, before canning or before bottling, if the food has already been contaminated. So you can have organisms like Clostridium botulinum that can thrive without oxygen in your canned food or your bottled drinks. Humidity and storage time. Moisture and prolonged storage encourage microbial growth, like cereals, grains, and flour, even though they don't get spoiled easily. But if the atmosphere, the condition, we usually have heat and then you have humidity in the air, so they might get spoiled. So you can see your flour caking, your cereals clumping together and changing color to yellowish or orange or green or black coloration. And your sugar can become watery. So what are the food preservation techniques? What techniques can you use to preserve your food? You can use physical methods, chemical methods, and biological methods to preserve your food. Refrigeration and freezing slows microbial growth, so they can be used to preserve food, and this is what we mostly use in our houses. Heat treatment, pasteurization destroys pathogenic bacteria, e.g. in milk and juices. When you do your IT next year, I think you will even start your IT in 200 level. If you are lucky to work in such industries where they produce food or juice or milk, you will discover that they do pasteurization a lot. After bottling, they pasteurize to destroy pathogenic organisms in the food. Then sterilization, that's still talking about heat application. Sterilization of food before canning will kill all microorganisms. Dehydration and freezing and freeze drying is another method, removing moisture to prevent microbial growth. Apart from the industries that we do this biotechnologically, in our houses, we do dehydration. When you apply salt to food, you render the organism, even though there's water there, the water will not be made available to the organism because of the excess salt. So the organism will not be able to spoil the food easily. The same thing when you smoke, you remove the water. When you sun dry, you are also draining the water so that you can prolong the food and prevent microbial growth. But in the food industries, they can also apply syrup. When you see your baked beans, you see that it contains syrup. The syrup will make the water unavailable to the microorganisms, so there will be no microbial growth. And because it has been sterilized, everything is intact. You have your sardine, it is overlaid with oil and it is salty. So this is to prevent availability of water to the microorganisms that might want to get it spoiled. The chemical methods include preservatives like sodium benzoate, nitrates, and sulfites, which inhibit microbial growth. Organic acid, like I mentioned, vinegar, that can be used to sterilize. Vinegar can also be used as a chemical method of preservation of food. Vinegar is simply acetic acid, and then citric acid that will lower the pH of the food, thereby preventing microbial growth. Biological method like fermentation. Fermentation uses beneficial microorganisms to outcompete pathogens. During the process of fermentation, the pH may be lower, so most pathogenic organisms will not be able to survive. So fermentation helps. When you ferment your iru, you can put it on the bench, nothing will happen. Bacteriocin is also a product from microorganisms. It's a natural antimicrobial that is produced by lactic acid bacteria, e.g. lysine, and they are used in the preservation of food. So you have food preservation techniques like physical, chemical, and biological methods. Let's look at some common foodborne diseases. We've mentioned some of them. Commonly, we have salmonella that can cause a disease called salmonellosis. What are the sources of this? Raw eggs, poultry like chicken and turkey, even meats. You also have E. coli. You have different strains of E. coli because it's not all E. coli that can cause diseases. They can cause hemorrhagic colitis. And you can obtain this organism, you can eat it with your contaminated beef or contaminated vegetables. Listeria monocytogenes causes listeriosis. This can be obtained from dairy products and other ready-to-eat food like vegetables, like salads. Clostridium botulinum causes botulism, and it is obtained because Clostridium is an anaerobe. So it can be obtained from improperly canned food. Maybe the can is not sterile. Maybe the bottle is not sterile before it is canned or preserved. And I mentioned earlier, Vibrio cholerae that can cause cholera. There are others, but these are the common foodborne diseases. So we must not finish this topic until we talk about food safety measures. So because we know that food can spoil, food can cause diseases, we have to ensure that our food is safe. So we must put in place some food safety measures, to have properly safe food for consumption. So one of the method or measure is good manufacturing practices, the GMP. Proper handling and processing of food is necessary. You have good manufacturing practices. For those of you that will do your IT in production outfits, food production outfits, or you later work there, you will understand what GMP is all about later. And there are basically 10 principles of good manufacturing practices. This includes standard operating procedure. There must be a properly written standard operating procedure. Like I was saying earlier, when you were asking me about thermophilic organisms, every industry have their own procedure. So the procedure is already written there, and it will be in the lab to guide whoever is working there to follow. A staff that does not follow the standard operation procedure, or what we call SOP, will eventually put the company into trouble. So you must follow written procedure, like written procedure for cleaning, written procedure for kitting, and all that. There must be proper documentation of what you have done. Validation, for example, if you are preparing something and it is time to close, you should be able to document that, oh, I have added this and this to this product, so that you know where to start from. Design of facilities and equipment must favor the safety of food. It must favor the safety of food. Why? Because you cannot have a toilet close to, for example, when you are producing your juice. The toilet should not be there, close to the manufacturing equipment. Maintenance is highly necessary. If you don't do a proper maintenance, the competency of employees, the group of people you appointed to do your food
Rachel,
production
mute your
in
mic.
the food industry, are they competent? Do they know the rules and regulations?
All right, please let me do that first.
Personnel and hygiene practices, very important.
Mute your mic.
The personnel working in food industries must be properly kitted. You go to the industry, you must wear your cap, your nose mask, your gloves, the lab coats, and then there's a special shoe they will give to you to wear. But what do you have in Nigeria? You see outside the lab, you see people coming out with their white coats, lab coats with the blue cap, and you see them moving about outside with the shoes that are supposed to be used only inside the lab and the factory. So by taking such shoes outside, they will bring some other microorganisms into the lab or the production room. So these rules have to be followed. Then the quality control, there must be a quality control at the point of production, at different points of production. The quality control officer must be able to take sample, the raw samples, when people bring raw materials, when suppliers bring them. The quality control officer, supervised by the quality control manager, must be able to take samples randomly, analyze it, and ensure that it suits their need. There was a case of, is it my Peking? At that time, maybe some of you were not born yet. There was this contamination of my Peking teething powder that was used for babies in Nigeria, and it wiped out babies that year. They discovered that it became a court case. They arrested the owner of the industry and also arrested the quality control officer. After thorough investigation, they took some samples to other labs, and they discovered that what happened was they used an excess of a particular chemical. For example, if they are supposed to put maybe 0.1 ml and the person accidentally put 1 ml, 0.1 is far, far less than 1 ml. So that was what killed those children in those days. So the quality control is very important. Somebody was careless along the line. There's supposed to be, before, when the raw material comes, you test it. Is it okay? Is it up to standard that you request? If it is not up to standard, you return it to the supplier. Then during the production in the industry, you take it at every point to analyze. Then until you get to the store, while in the store, before you give these items out to consumers, you also test it from time to time to ensure that it meets the quality before you give it out. Then there must be auditing and compliance. So apart from the good manufacturing practices, you also have the hazard analysis and critical control points, the HACCP. In this case, you identify and control food hazard. And I have a diagram here that shows HACCP and what it means. H means hazard, A means analysis, C means critical. The other C means control, and P is points. Hazard analysis, critical control points. And you can see hazard is whatever that is dangerous to health. Analysis is to investigate the hazard, if you have any hazard. Critical means it is very crucial for you to contain that hazard. Control, you must handle the condition that is causing this hazard. Then the points, you must identify the position, at what point this hazard occur so that you can control it. For example, you are producing water. A critical control point is your filter. You must take your water sample for analysis. After it passes through the filter, you want to know whether that filter has done what it's supposed to do. So there are critical control points along production line, like washing. After washing, you take sample. Filtration, you take sample. You put it in the UV light, you take sample. You analyze, you see whether every point is working the way it is supposed to work. If it is not working, you check. That is where maintenance comes in. You check, you document, you call who is supposed to do the maintenance, you repair, then you go back again until you have the right result before you release the product. Proper cooking and storage temperature is another food safety measure. So cooking at high temperatures and refrigerating perishable foods will help you, to maintain the safety of your food. So in conclusion, microorganisms play a dual role in food systems, contributing to both beneficial and harmful effects. Understanding the microbial ecology of food helps you in ensuring your food safety. Developing food biotechnology, because when you know the problem you have, you want to look for a way of solving this problem using microorganisms, like in food biotechnology, and preventing foodborne illnesses. Proper handling, processing, and storage of food are very critical in controlling microbial growth and protecting public health. So we come to the end of the lecture on the role of microorganisms in food. Any question? So we've had lectures for over 2 hours. Sarah Ayorinde, I can see your hand up.
Yes, ma'am. Good morning, ma'am. Ma, please, oh, sorry. Good afternoon. Is it possible we use lime to sterilize some of our food? Because
yes,
I feel like
lime, lemon, vinegar, they are all acidic.
Okay, so we can put vegetable in a bowl and maybe just pour lime
In
water.
fact, I'm not saying just pour lime. Sometimes you even want to eat salad. You sprinkle some lemon. Just squeeze some lemon. Apart from giving it a good flavor, it's also help you to destroy some pathogenic organisms. Vinegar is very good. It's just citric acid. So you can soak your vegetable in it before you prepare your salad for a few minutes. If you don't have money for vinegar, you can use salt. Just soak it in salt water, especially ready to eat food like salad that does not require cooking or heating. So you sterilize with salt, with lime or with vinegar. Then you eat, you rinse, and then rinsing again, meaning you are rinsing with clean water, and that your own hand that you are using is also very clean. Sometimes you are cooking and you are pressed to go to the toilet. Go to the toilet, some people will just rush out. They will not remember to even wash their hands with soap and water. All right. Yes. For ready to. Sometimes, when you have slimy food like catfish, if I don't have vinegar, I use lemon or lime. I just sprinkle. When you have slimes on such fish, that means you have microorganisms. If you put a bucket or a bowl of water in your bedroom and you travel for a week or two, when you come back, what do you feel? You want to rinse the bucket and it is slimy. Those are biofilms of microorganisms. They have survived and they have attached themselves to that bucket. So you need to get rid of it by first washing it, then maybe using Dettol, to disinfect. The same thing with something like fresh fish, like the one that have scale is very good. Scaly fish are the best. Because as they survive in water, different microorganisms, salmonella, whatever is in that water, depending on the river. If it is river, like the lagoon where people defecate and urinate and throw, even human beings are dead there, you have to sterilize. But the scaly fish, there's no problem because you remove the scales. So once you remove the scale, it is the scale that have housed the microorganisms. So once you get rid of the scale, for example, croaker fish, you remove the scale and your fish is safe. But for catfish, you need to, decongest those slimy microorganisms that have over time attached themselves to it. So you can use vinegar, you can use lemon. Once you just sprinkle lemon in the water containing the fish, you will discover that the slimes are coming out. Then you do that severally. You just discover that the fish is hard. There's no more slimes. That means you have sterilized it. Then, in addition to cooking, you have gotten rid of the organisms. So I cannot go to a party, for example, and eat asun fish. I don't. Maybe because I'm in microbiology. So when people are fighting to get asun fish or asun chicken, you can't find me fighting over it. If I have the opportunity to take it away, then I'll go to my house and put it in the microwave or put it in the oven to further heat it before I can then consume. Did I answer your question?
Yes, ma'am. Thank you.
All right. Amen. Olamide Miriam.
Yes, ma'am. Thank you, ma'am.
Yes.
You said that there are microorganisms in the freezer that can survive for cold temperatures, right?
Yes. I didn't say I didn't say there are microorganisms in the freezer. But microorganisms can survive the freezer. It is not the freezer that brought about the microorganism. It is your activity of putting food, bringing food out and keeping it and not cleaning your fridge that allowed the microorganism to persist there. So when you even put your food, after 5 or 6 days, it will still get spoiled.
Oh.
Yeah, because the organism will be able to, though the temperature of the fridge is slowing down their growth, but eventually they will still grow. But when
what if it? Because some of us, we like to put something like beans in the freezer for 6 days or 5 days.
Freezer is okay. Freezer will stop the activity of the microorganism. Okay. The problem we have with Nigeria is that the electricity is not stable. So you put your food in the freezer, it is almost freezing. Then they will take light for another 9 hours. And it's like that every day. It's even more dangerous than not even putting it in the fridge at all. So you have to study. Do you have steady light before you begin to say you want to use that method?
If you continuously heat it, can it not eradicate the micro?
Last long. Demilade.
Oh, foolish question now.
Demilade. Okay, since Demilade is just raising his hand without being attentive. Yeah. Please, I cannot hear you. Your environment is so loud. Too noisy, please. If I have another person, let the person go on. Otherwise, I'll call it a day. I can't hear you. You are cracking. Adeuja. Your internet is bad. I cannot hear you, please.
Type the question. So type it in the chat box.
Hello, ma.
Hello.
I have a question, ma.
Good. Who is talking?
It's Angel.
Angel. All
yes,
right.
Ma. There are some countries, or yes, let me just say countries or some continents that they eat like raw meat. There's a particular raw meat they eat raw. They consider it like edible. So raw meat. So I want to ask now, is it that that's not that for them to consider it edible?
Anyway, well, the noise is so much at your end. I can hear car on and all that. But if I get your question correctly, you said there are some countries where they consider raw meat as fit for eating. Like countries like Japan and all that, they eat sushi or whatever they call it. Yeah, it looks raw, but it's not entirely raw. Most of the time, it's half cooked. And the one you consider raw has been soaked, has been sterilized using chemical methods. Is that clear?
Yes.
Any other person, please? Any question? Real one? Are you still there?
Yes, ma'am. I'm there,
okay, so real one, I want to appreciate your efforts in trying to coordinate the class. God bless you. So please, you know, so many activities going on. If you don't see your notes, if you don't see your slides by evening, please remind me.
No problem, ma'am.
Yeah, so I'll share the slide on our platform so that everybody can have it, and those who miss the class can also read it. Thank you. Ovoke, yes, are you raising your hand?
Yes, ma'am. Good morning.
All right.
The question in the chat box.
Use alum water or to use it to.
I can't hear you. You are breaking. Okay. You are breaking. Speak again. Let me see.
I said, ma'am, I wanted to ask about the use of alum.
Alum in water?
Yes, ma'am. Like, what is it?
Alum in the water factory is a method of coagulation. It is not sterilizing the water. In the process of water packaging or water treatments, at a certain stage, you use alum to coagulate whatever you have, including they will clump together. You see that when you put alum in water, you have particles that are clogging together. Then you might now go further to filter. After filtration, then the water may be fit for drink. Did I answer your question?
Yes, ma'am. Thank you so much, ma'am.
And it does not end there. It depends on your raw water. If it is sewage water, you use alum, you coagulate, you filter, but you still go ahead to sterilize using UV, using ozone, using other, there are several other methods that you can use. But if it is tap water, the method you will apply in the sterilization of water depend on the raw source of the water itself that you want to sterilize. Do you understand? If I take sewage water, it will take some different method to be able to bring it up to drinking level. Some people recycle their water, whether it is from the toilet or from, it's recycling, and they bring it up to the level of drinking, but they have to apply several methods. So lagoon water will require more time, equipment, energy, and different methods to bring it to drinking level compared to tap water or rainwater. I hope you can understand what I'm talking about.
Yes,
okay,
ma'am.
All right. So Adeuya Lemilade said, Ma, recently I have seen that people use baking powder to wash the back of pots because of its ability to clean them, but use the same product to bake. Yeah, that's using baking powder to wash pots is not my business here, but baking powder is definitely used to bake because it has some microorganisms like yeast that can help you to rise your dough, to make your dough in donut or puff or bread to rise. But by using it to wash pots, I don't know anything about that. When I want to wash my pot, I don't even use suits in my house, so I don't know. I know there's scrubbing powder for people to use, so I don't know about the use of baking powder to wash pots. Baking powder in this subject is used for baking. Any other question, please? All right.
But ma, for holy water, the best way to like purify it is just boiling. It's like simplest and best for drinking.
Boiling of water.
Just boiling. Just to boil the water.
Not just boiling, boiling and filtration, please.
Okay.
You know that some organisms can survive 100 degrees centigrade.
Yes.
Yes. That is why when we want to do absolute sterilization in the lab, we use what? Autoclave. But yes. you cannot be autoclaving your water to drink. So if you cannot buy bottled water or packaged water, you can boil. Then after boiling, you filter. That is what our mothers in those days does. They filter it and then you drink. Because there are filters. When you have filters that are below 0.2 microns, it will actually capture all bacteria. It won't allow them to pass through. So they'll be safe. You can boil and just filter, and that is fine. Any other thing, please?
Well, can we get the recording of the class?
I will try to look at, instead of me to even allow it to go straight to live. I don't know. I should have even recorded it on my computer and then send the recording. But now I did record on the computer. I said live on YouTube, and I don't know why people cannot see it. I'll try to go to my YouTube page to see if I can get it. If I can get it, then I'll send it to the platform. Otherwise, I will just send this slides, and then I will try to look at what has gone wrong. All right, but even if anything has gone wrong, I will always record on my computer so that I can give the recording, the video recording later, so people can listen to the lectures over and over again, and also get firsthand questions that have been asked. So there will be no problem with that. I will be coordinating this with real ones, so there's no problem. Oh, okay, your hand is still up. Are you okay?
Yes, ma. I just remember the question, ma. Just now. So sorry, ma. Though it's kind of funny, but I'm just inquisitive. So since it goes in alignment with what you taught us today, so the yeast you mentioned earlier that is being used in I think used for baking. So sometimes I just think, just like the normal layman, like I just think that okay, those yeast I usually talk about, is it that they are moving new or they cultured like those yeast? So and now we add them to the baking, ingredients that makes it rise, or like is it they were being cultured before you can just, because I'm just like, how would they use yeast? Like yeast?
Yeah.
So it's But
When I was growing up, the kind of yeast that we use for rising the dough for our puff puff is like, it has the color of something like hash color. It is round. They just scoop maybe 1 teaspoon. There's already instruction. Depending on the quantity, you just scoop it, soak it in water, and within few minutes, you see it puffing up. You mix it with your flour, and then you cover it with nylon or something for like 1 hour or 2, and then you see it's already risen. Baking powder is also a kind of made from yeast or their enzymes, and they have been packaged. No, you now have all these things now have enzymes even in your washing detergent. You see a detergent, but there's enzyme there. That is green detergent. It is not chemically harmful to the environment. It is the enzyme that has been obtained from microorganisms and mixed with some powder as carriers, So you are seeing the baking powder as white, but there are enzymes that are from yeast there. So sometimes they synthesize, or sometimes the yeast may be directly from the yeast, the microorganism. It's not all yeast, all organisms that are edible. You have different species of mushroom. You've heard of people eating mushroom and the whole family dies. People have, I don't eat mushroom. My mother eats mushroom, but they have they have a way of identifying the edible mushroom and the poisonous mushroom. So I don't do it. They eat it all over the world. And mushroom, even in the developed country, they cultivate mushrooms in the lab. They have industries where they produce edible mushrooms and people eat. And it is proteinous and very good for the eye, but I don't like it. Any other stress, please? It's not easy talking for 2.5 hours. Yes. In the absence of that, we'll meet on Friday. I will check the time up with Real One. I will try to see what we can do because I want us to finish latest Tuesday or next week Friday. That is why I have done some sleepless nights to produce all these slides to make work easier for me. So I don't keep postponing my lectures. So I won't be able to make it today or no. We have to finish so that you have sufficient time to read while exam is going on. I'm the exam officer for microbiology department. So I have other exams to take care of from 17th. So I must finish your lectures before 17th. I hope everything is okay that way, right?
Yes,
Yes,
ma'am.
All right. Thank you very much. So please enjoy your day and God bless you. Yeah,
Thank you,
bye.
ma'am.
Thank you, ma'am.
Bye
Thank
bye.
you, ma'am.
Have a
Thank
nice
you.
day. Please help me talk to your colleagues to always mute. To mute. Most of them, they just want to mark register. They are not listening to any lectures. Why? There's no crime. It's not, we don't expect that, okay, 100% you must attend class. The rule says 65%. So if you have to be absent 1 or 2 or 3 times, it's okay instead of just disturbing the whole class. Please talk to your colleagues. Thank you.
Thank you, ma.
Yeah, bye.
Thank you, ma.
Bye, ma'am. Thank you, ma'am.
Bye, ma'am.
Bye.
Bye.
Bye.